I'm Lieutenant Colonel Sylvain Hubac. I'm the head of the DNA unit at the Forensic Science Laboratory of the French Gendarmerie, and I'm going to present the using of RHID technology in my lab. So the title of the presentation is Rapid DNA at the French Gendarmerie, a mobile and remote control applications for decentralized human identification capabilities. The plan is the following. For beginning, I'm going to present the validation study for reference, casework, and DVI samples. After presentation of our overseas project using RHID system and two different applications, a mobile application and a remote control application. And at the end, I'm going to present implementations of the technology in progress. So first, regarding the validation study for reference, casework, and DVI samples. So we did a comparison between the result obtained with our routine workflow compared to the RHID system. And for that, so of course, we use a RHID system, and we use two different types of cartridge, so ACE and or Intel cartridge. And the interpretation of the result was done using GeneMapper. For the routine workflow, we performed DNA extraction using Adentech crime prep kit. We don't perform DNA quantification because there is no quantification using RHID. So in order to compare, we didn't do that. DNA amplification was done using global failure kit, and the genotyping was done using a 3500 XL. And for the interpretation, we used also GeneMapper IDX. Regarding the samples, so we analyzed different types of samples: buccal samples, cigarette butts, blood, muscle from fresh cadavers, bones, bone marrow, and touch DNA for a total of 120 samples. Regarding the samples in details, so for buccal samples, we use 11 different individuals, and the collection was done using flock swabs. For cigarette butts, we use 3 different individuals, and we collect only 5 millimeters of filter paper and not the filter for a total of 10 cigarette butts. For the blood, we used only 1 individual, and we performed serial dilution 1 to 1, 1 to 10, 1 to 100, 1 to 150, and we spot 10 microliter of blood on a flock swab, and we did 8 replicates per dilution. For touch DNA, we put 3 fingerprints spotted simultaneously on a fragment of glasses, let dry 1 day at room temperature, and the collection was done using a flock swab. For the muscles, we used 2 fresh cadavers, and we did 6 samples from psoas, and it's a little bit, it's a piece around 2 millimeters collected. For bones, we collect bones from 3 cadavers, 2 femur and 1 rib, and the collection was done using a specific trypan patented by my lab. And with this trypan, we can collect cylinders of bones, and cylinders around 20 and 100 milligrams. And at the end, for the bone marrow, we realized 9 collection of bone marrow inside the femur using a flock swab. Now for the method, so for each type of samples, the first half replicates is analyzed using RHID, and the second half is analyzed using our regular workflow. For buccal samples and cigarette butts, we used ACE cartridge, and for blood, touch DNA, muscles, bones, and bone marrow, we used Intel cartridge. Now, for interpret the result, we use a color code. So there is many colors, like you can see. But the most important to understand is if we have a green flag, that means the efficiency between the RHID technology and the routine is similar. For example, if we have a blue flag, that means the result using RHID is better than the result obtained with routine. At the end also, we can see here that there is a green flag if at the end the profile is interpretable, and we consider a profile interpretable if we can obtain 2 alleles for more than 6 markers, and if we are behind this this number of loci, so that means the profile is not interpretable. So for Buccal samples, we obtain 100 full DNA profile and no difference between RHID and regular workflow. For cigarette butts, we obtain better results using the routine workflow. That means we need to improve the quantity of samples, perhaps, and not only use a paper filter, but also use the filter. We analyze some samples, cigarette butt samples, using paper and filters, and we obtained some troubleshoot, some technical troubles when we did that, because for different cartridge there is a problem to the liquid to go through the cartridge, and we don't obtain a result with this problem. Another point to improve the result from cigarette butts, it's perhaps to use not ACE cartridge, but Intel cartridge, because when we use Intel cartridge, the number of PCR cycle is it's higher, so you can obtain a DNA profile. I think we can obtain a better result. For blood samples, so there is no difference between RHID and routine until the dilution 1 to 50, but the results are better for the routine because for higher dilution, because we obtain a full DNA profile until the dilution 1 to 150 with the routine workflow. For muscles and bone marrow, so there is no difference between these two technologies. And for bones also, for fresh bones, there is no difference, and we obtain an interpretable DNA profile from each cylinders of bones analyzed. For touch DNA, the results are better using the routine workflow because only 2 samples from the 10 samples analyzed gave a good result. So in conclusion, regarding the validation study, the rapidity is not validated for touch DNA samples. The throughput, 1 sample in 90 minutes, it's a little bit slow. So that means 1 to 5 samples in 90 minutes simultaneously and independently could be better. And one limitation is there is no internal control to explain a negative result. But I know that this type of control is on the pipeline of Thermo Fisher with a new cartridge using a global filer. Another limitation is it's a one-shot analyze. We did some tests to reuse buccal swabs, and we have demonstrated that it works. But for casework samples, it's more challenging. So we find an issue to that, and we performed a regular DNA extraction from the sponge, so from the sponge at the back of the cartridge. And like you see in the graph, so we obtain higher quantity of DNA, so higher intensity of RFU on the profile if we perform DNA extraction from the sponge. The system is also very sensitive to micro blackout, so that means you need to be connected to battery or inverter to prevent troubles. In conclusion, so the system is very easy to use and compact. This is a good point. And we have validated this technology for rich samples. So that means saliva, blood, fresh bones, and also for DVI context. And we have demonstrated that you can use only one type of cartridge for all type of the samples. So we have retained Intel cartridge. So after this validation, the project was to develop a mobile and a remote control application. So why? Why? Because in France, we are a metropolis of 65 million of people, but we have also 10 overseas with 2 million of people in 3 different oceans. There is a high level of criminal activity in France. So, for example, per day, there is an homicide each 8 hours. There is an assault each 2 minutes, a sexual assault in 10 minutes. So there is very high level of criminality. There is only 6 law enforcement agencies, so 6 labs in France, 5 for police and 1 for gendarmerie, located around the country. And there is 10 private labs. But there is no lab or private lab, no public lab or private lab on overseas. Another point is that the volume of DNA samples per year that we perform in my lab is 55,000 for caseworks and 145,000 for reference samples. And the volume of DNA samples for overseas, including Corsica, is around 5% of caseworks and 10% of reference. That means there is a real need in overseas to perform DNA analysis. But there is no lab. And when we consider the flight duration and the jet lag, it's very complicated. That means there is no DNA analysis in reduced time possible and compatible with the examination time of a suspect. Just for an example, if we consider New Caledonia, so New Caledonia is here, Paris is here, so there is a flight duration of 24 hours and a jet lag of 10 hours. So that means there is a real need to provide DNA analysis capability in real time for sensitive cases in overseas. But there is some problems, and the limitations are due to the French law, because for the French law, DNA analysis must be done and validated only by DNA expert accredited. Accreditation is delivered only by the Ministry of Justice, and it's difficult to obtain accreditation, even more difficult when the technology is new and considered like a black box. At the end, technology must be certified ISO 17025 by the French accreditation organism named COFRAC. But now all the limitations are fixed, and RHID is ready to go in France. That means the technology is really validated, but restricted to rich samples, like I said, saliva, blood, fresh cadavers, fresh bones. We obtained certified by Ministry of Justice since June 2021, and so that means we can, the result gave by the RHID can be used for national DNA database for reference and casework samples, but it took 18 months to get it. Another important point, and we are now certified ISO 17025 by our state accreditation agency since few days, and it took 18 months also to get it. There is 2 applications that we want to do with the RHID. So there is a mobile and a remote control application. For the mobile application, DNA experts, so DNA experts are here, IRCGen are in Paris, they go to the field with the instrument and they process DNA analysis. And for the remote control application, DNA experts stay in Paris and analyze is decentralized. So that means only the machine is in the field, and she's used by a local technician under real-time control by a DNA expert in Paris. So for the application, if we have more than 5 samples to process on site in DVI or blood crime scene and reference samples, and when there is also touch DNA samples to analyze, so we don't use RHID technology and we use our mobile DNA lab where we can process our routine workflow inside. If we have only 1 to 5 samples to process, so for DVI, blood crime scene of reference samples, so we don't use our mobile lab, but we use a RHID mobile desk. And so what is this mobile desk? So it's an all-in-one flight case that we developed internally. And inside this flight case, there is a fridge for consumables, there is a battery, there is a solar panels. So we can see on this slide the flight case. and at the back of the flight case, there is a power supply. That means that the instrument still running inside the case during the transport. If we open the flight case, so you discover inside the RHID, the laptop, the front panel can be used like a desk, the fridge, and a solar panel in order to obtain a full autonomy even if we have to go on field without power supply. There is also a battery, and with this battery you are 48 hours of autonomy. So using it, you can go wherever you want in each point of the world without stop the machine. So this is for the mobile application, and for the remote control application, there is a list of requirements. And so only the instrument and the laptop are installed in local forensic lab of the French gendarmerie. So you can't install the instrument outside a forensic lab of the French gendarmerie, and we have a forensic lab in each department of France. The local forensic lab must be certified ISO 17025. The instrument is still the property of IRCGN. The instrument is managed only by IRCGN, and the training of local technicians is done also by IRCGN. Local technicians, they don't have access to the laptop. That means they don't use a RHID link. They can't see the result. And the local technician must just log in the RHID, and we also control the face of the technician by using an internal camera of the RHID. The technician just to scan the barcode of the samples, and also we can control the samples by RHID camera. Insert the sample inside the sample cartridge, insert the cartridge in the RHID, remove the cartridge after the run, and that's all. Data transfer from the local forensic lab, so that means, for example, in overseas, to IRCGN, so in Paris, is done using an internal secured network. So the details of the secure network is presented on this slide. So basically, this is a computer in Paris, and on this computer, we have tight VNC application, and using tight VNC, we can connect directly to the laptop of the RHID in overseas, and by using RHID software, we can connect directly to the instrument. So concerning the data transfer from the RHID laptop to IRCGN workstation and uploading of the result on the DNA database, so basically it works like that. So we use the computer in Paris. We use WinSCP application, and with this application, we can have an internal secure network to the computer connected in overseas. And by WinSCP, we have access to the folder where the data generated from the RHID are stored. And we have just to drag and drop the file from the right to the left in order to transfer data inside the secure network in Paris. And we have two possibilities. The first one is to copy and paste all the run folder generated by the RHID, or just the ladder and the sample generated by the RHID. And if we consider just the ladder and the samples, the weight of the file is very little, so 200 kilooctet, and that means the time to transfer is less than 3 seconds. So it's very quick. Until the transfer is done, we interpret the result in Paris by the DNA expert using GeneMapper, and we have just to upload the result inside the DNA database. Now, concerning the implementations in progress, so for the mobile desk application, we use it for a bloody crime scene. So it's a crime scene where there was very high quantity of blood, like you see in this slide. And so we collect different stains in order to define the scenario of the crime, because for this caseworks, many people were involved on the crime, and many people are died. So we obtain many stains of blood on site. And so basically, directly on time, we collect the blood with a swab, and we put the swab inside the machine, and we obtain a result in less than 2 hours. So by this example, we have obtained 3 or 4, I remember, 4 different DNA profile, and it will be very easy to identify the victim. For the decentralized application, so the first implementation on overseas is in New Caledonia since October 2021. So remember, the flight duration to reach New Caledonia from Paris is 24 hours, and the jet lag from Paris is 10 hours. And we obtained the first hit in November, so just 1 day after the end of the installation. And so it was very good opportunities for us to demonstrate that this kind of capabilities in overseas could be very interesting, because in just few hours after the murder, we identified the criminal. For the implementation, we plan to install a second instrument overseas, so in Guadeloupe, perhaps end of 2021, depending the situation for the COVID, and also a third site in Mayotte at the beginning of the next year, and depending also the situation of the COVID. So if you have any question, I can answer with pleasure. Thank you.